Welcome, everyone. We'll still take some time. We'll start on top of the hour, but give me a second. I'm posting the paper here on top of the room so people can have a look at it while we are waiting. And our guest speaker will give soon a TED Talk actually about his research and this topic. And this is kind of a nice preview of the TED Talk that will be happening. So yeah, there's the paper. Feel free to check it out. It's really interesting. And I'll hope. Yeah, and if you think of people that that would like to learn more about the environment and yeah, feel free to share the room. Thank you so much. And Dr. Averill, he will be joining us from Switzerland and Zurich. Hi Manas, how are you? I hope you like this room. Hi Jonathan, and hi Christina, David, Gunvald, Nisin, Nishk, Siva, Mayor. Welcome everyone. I think this topic is really important. It's kind of a upcoming TED Talk preview from Dr. Averill. And yeah, the paper is posted on top. Hope you enjoy it, and I'm looking forward to learn something from him. So, and I'm glad he could make it. He's right now really busy to try to. He has a foundation to protect the microbiome of the earth, especially different fungi that are kind of being threatened. And he is currently in the very busy finding funds, but he still made time for coming here for an hour, so I'm really glad he did. And will be really interesting. Thanks, Jonathan, for sharing the room. And we'll start on top of the hour. As usual, I just usually open up the room a little bit earlier because usually our guest speakers are new to Clubhouse. And just to give them a little bit of time to log in to come here, just in case they want to try things out first. I'm always here a few minutes ahead of time. It's almost Thanksgiving. Happy Thanksgiving. I hope that's okay to say it ahead of time. I don't know these rules. And just to give a little bit of a preview, so because it's Thanksgiving, we won't have too many rooms, and last week I was traveling, so we only had one room. So this week we have, if you go on our club and check the calendar, we have two rooms, today's room, and then on Friday again, Dr. Djakovic will be talking about coexistence of modern humans and Neanderthals. His group published very recently a paper that they found that humans and Neanderthals coexisted quite closely, way closer than we thought before, for a long time, based on recent findings. I think that's really interesting because a lot of our genome is kind of intertwined with Neanderthal genome, and it has good sides and bad sides. And I think the more we learn about that and of our past, the better. So I think this will be really exciting. And then next week, Monday, we'll have Dr. Sergi, who did really interesting research on black widows and how they have a really good spatial memory of where they captured their last prey. That is really interesting because, this really precise spatial memory was shown in mammals, but not necessarily in non-mammals before in that way. And to do that in spiders is really not that easy. It's a pretty different way, of how the brain is structured. So that was quite amazing. I had to invite him. And then we'll have Dr. Feldman talking. He publishes and works a lot on meditation and how it affects the brain, and studies that for many years now. And he will be talking about that and how breathing patterns influence emotion. I think this will be really interesting around the holidays, that we learn about. The holidays can be great and they can be stressful. So I thought that was a great way to kind of have a room talking about stress, emotions, and how breathing patterns can change them. So yeah, this is the preview for this week and next week. And we have a little bit of a lighter. Usually we have like 3 to 4, 5, sometimes even 5 rooms a week, but around the holidays we'll have less rooms. And then in the new year, I have already a few rooms in the new year, and even a speaker that requested a spot in March, but it's still a lot that I will invite, depending on what will be published in this and next month. I try to stay as as new, like the knowledge comes in, like when something gets published. Hi Colin, how are you? Can you hear me?
Yeah.
Ah, perfect. How are you today?
Hi. I'm doing good. How are you?
Good. Thank you. We're just talking about our upcoming program. I usually open the rooms like a few minutes ahead of time to give people time, to arrive, like especially the speakers if they're new to Clubhouse and so on. So I was just talking about upcoming rooms and about your work.
Awesome.
A little bit, just a little bit, to not give too much away. Well, people will be coming in, and we record the rooms, just to let you know if that's okay with you, because our members are really truly around the world, so there's never time that works for many people. So, the only way to solve this is to record them. And here on Clubhouse, the cool thing is when you listen to the recording, here the links that are available, like if you see on top of our icons, there's the link to your paper. They are still active, and also the chat is active because we usually share a lot of additional resources in the chat. Like it's all still active, so it's kind of almost the same experience. So that's why we like to do this on Clubhouse. So I hope that's okay with you. If not, I can turn the recordings off. But yeah.
No, that sounds great. And thanks so much for organizing. And thank you for dealing with me having to reschedule a few times. Yeah.
Yeah, of course. Yeah, we are honored having you. And yeah, it's a pleasure. I'll give a short introduction, and then I'll ask you a couple of interview questions, if that's okay with you, and then we go into your research and
Absolutely.
perfect. Okay, so welcome everyone to Science Society, and of course, special welcome to Dr. Colin Averill, and he is at the Crosser Lab at the ETH Zurich, and what the team works on different projects that are around the forest microbiome, ecosystem, ecology, soils that are alive, and the diverse forest microbiome communities, and the profound impacts on the world and our world that these microbiome communities have, and how this diversity affects the trees, the forest, and carbon sequestration. That also then affects climate change, and how the diversity and population in the soil can maybe also give us a forecast of climate change. In special, Dr. Averill is focused on the fungi in the soil, and I think this is such an interesting and really not well explored research field so far. So our first question is, how did you realize you were interested, or you had like this passion to go into the field of research in general? Did you always want to become a scientist, or was it something that came up later? Maybe you took a class or you read a book. What kind of pulled you into this world? Thank you.
Yeah, totally. Thanks so much for having me. So I think I really got my start because I was trying to fulfill a biology requirement for another degree I was thinking about pursuing, and I took an ecology course, and I could not believe that this person instructing us like studied butterflies for a living. I was like, that is insane. You're an insane person. How do I do this? But I eventually got really excited about the role of microbiology, so all the bacteria and fungi and other organisms that live in soil, and the role in climate, through a professor where I did my undergrad at Boston University. I was washing dishes in a terrestrial ecosystem ecology lab, where they studied the forest and how the forest works. And I learned that there was like two major uncertainties in climate science. Like, we don't know how clouds work, and we don't know how long the land surface, so all the forests and grasslands and other ecosystems on Earth, how long will that system be able to continue removing carbon dioxide from the atmosphere? And I didn't know anything about clouds, but I was much more excited about forests, so I got started there. And then as I got to know that space better, I learned in these models that represent how a forest works and our best understanding, the thing we knew the least about in those what was happening in the soil, and particularly with all the things that live in soil. And we had no way to measure it, but knew it was massively important, one of the biggest uncertainties in the terrestrial carbon cycle. And that's how I got excited about it. I was like, this is where we need to focus. And so um, I think that was really what triggered me, and it still motivates me today.
Yeah, that's fascinating. That's also an interesting story that you found that really cool that a person can make a living by studying these things. And I'm oftentimes had also this kind of feeling that I was getting paid for doing a hobby um, at some point. So I kind of kind
Yeah.
of get that, and I really yeah. And then the next question would be for this specific, paper, and then also I know that you're a co-founder of the Spun Society for the Protection of Underground Networks. Um, is there maybe a background story around that, like how this specific paper came about, but also maybe your foundation um, or the organization? Is there like was it hard to get it off the ground? Were people very excited, or are people very excited about it? And is it easy to get funding for, let's, if you have something around that. Thank you.
Yeah, absolutely. So yeah, I'm the co-founder of a nonprofit called SPUN, which stands for the Society for the Protection of Underground Networks. It's focused on the conservation of mycorrhizal fungi. So mycorrhizal fungi are these fungi that form a symbiosis with the roots of plants. They're in basically every ecosystem on Earth. There's actually evidence when plants first made the evolutionary transition from living in water to living on land, they evolved this partnership with their fungi and their roots before they even evolved roots. And so this symbiosis is really ancient, and it's fundamental to how basically all plants access growth limiting soil resources. There's incredible biodiversity of these fungi all over the world. So the fungi you find in, pine forests are going to be wildly different than the ones you find in an oak forest or in a temperate grassland or in a mangrove. But we also know that this biodiversity is actually increasingly coming under threat. That's driven by large scale pollution we put into the environment, like nitrogen deposition. That's driven by land use change, us converting ecosystems from natural systems into really intensively managed ones. And so we were motivated to found Spun by that. There was this sort of hidden extinction going on before our eyes before we even really had a chance to really document this biodiversity and understand where it was. And the motivation for this paper was to really make that point more broadly. It's not just mycorrhizal fungi, it's all the organisms and the microbial life that lives in soils and on and inside plants and animals. All of this is incredibly important to the health of people, the health of plants, the functioning of soils. um, and we're only now just beginning to understand what's happening. um, a big part of that I want to emphasize is also, we really just never had the tools to look at this before. So people had studied soil biology in a lot of different ways, but fundamentally it was hard. And all of that changed about 15 years ago when environmental microbiologists started applying the tools of the Human Genome Project, so these next generation sequencing technologies that made that possible, and using them in the environment, and started documenting all the biodiversity that lives in soil. It was the first time we could really sort of turn the lights on below ground and see, the variety of life that lives there. And what we saw was astounding. The microbial biodiversity in soil is outrageous. There's easily 1,000 coexisting species, microbial species, in a single handful of soil. And only in the past, 5 years has that DNA sequencing technology become really cheap enough so that we can look at the soil microbiome at the kind of spatial scales we really need to understand, a global picture of the biodiversity that lives in soil. So all of these things sort of came together at the right time. And as we put together those pieces of information, what we're learning is actually a lot of it's massively under threat, from things like, converting forests into intensive agriculture, clear cutting forests, etc. So I hope I answered some of your question. There were a lot of parts, but happy to go back in any part of it.
Yeah, that's wonderful that you and your team members created this organization, and because so much is unknown, I would imagine were many people advising you against doing it? I can imagine that since it's such an unknown field, or maybe the opposite. People were totally like, was there anyone giving you advice that was against or for it?
Um, Maybe not directly advice, but there were definitely lots of signals that I should be focused on something else. So I spent a lot of my career has been spent in rooms with, ecosystem modelers and climate scientists trying to understand, why don't we represent these key components of biodiversity in the models we build to simulate the capacity of forests and other ecosystems to act as carbon sinks if they are so important. And so really trying to make that clear, understand why it hasn't happened, and what information that larger community needs to incorporate uh, and appreciate these components of biodiversity. And so through that process, I met a lot of people working on plant physiology and community ecology, and lots of things directly adjacent to microbiology in the environment. But it really was a process, and has been a process for environmental microbiologists to really bring this aspect of biology to the forefront of people's minds when they're talking about climate modeling, when they're talking about ecosystem restoration, when they're talking really about biodiversity conservation writ large.
Yeah, I agree. It's so important, and there's, I don't know, after forest fires, people attempt to replant things, but I guess the soil microbiome diversity probably until now got really ignored, and so yeah, I'm really glad you're here and that you will educate us about this. So I posted the link to, like, a link to the paper PDF that everyone can access on top. And yeah, thank you so much, Colin, and the stage is yours.
Awesome. Yeah, so I think to start, it's really too important to just dig a little deeper to understand what microbes are, and really what they do for us, as people, and what they do for the broader biology, plants and animals. And so microbes actually are in many ways the majority of Earth's biodiversity. There are millions of species of microscopic fungi. There are billions of species of prokaryotes like bacteria and archaea. These organisms were the very first life forms to evolve and inhabit our Earth, and they'll probably be the very last. And so we find microorganisms and microbial life in nearly every habitat on Earth. They live in the atmosphere, they live in and on plants, they live in our bodies where they have profound effects on health, they live in animals' bodies, and they completely dominate the soil environment. These are the organisms that do decomposition, and they power really important soil functions like nutrient cycling, which in turn allows productivity on land to happen. The healthy functioning of your body is really dependent on microorganisms, as is the healthy functioning of plants. And they represent this incredible variety or biodiversity across the planet. So, you know, if you go to one forest and look at what's living in the soil, and then you walk 100 meters away, you can find a totally different community of microorganisms. They're just that heterogeneous and diverse. But as we study this component of biodiversity more, we're coming to understand it's under threat, just like macrobiological diversity. So just like we're seeing declines in plant and animal and insect populations, it seems that we're also, you know, seeing declines in microbial taxa as well. So this was first really documented, as far as I can tell, by mycologists and amateur sort of mushroom hunters. So mushrooms are really interesting because they sort of live this dual life as both a microbiological organism and a macrobiological organism. But the mushroom you pick is just the tip of the iceberg, and below that, it kind of just dissolves into this unseeable but huge mycelial network. So the mycelia is like the body and the roots of a fungus, and most of it lives underground. But you see this little macroscopic signal of it when it puts up a mushroom. So people have been documenting mushroom biodiversity for really over a century in Europe, and they were some of the first to show that, wow, there's this incredible decline in the biodiversity, the number of species we see coming up year after year at a continental scale. And later, ecologists would discover that a lot of that's linked to nitrogen pollution. So when we burn fossil fuels, we emit CO2, but we also emit all of this sort of nitrogen gas that eventually rains down as fertilizer and actually decimates the communities of these mushroom forming fungi. And so this was really the first evidence, and things have changed now that we can see the soil microbiome in its entirety using DNA sequencing. And so this really gets to the first part of this paper. We break it into 3 sections, and the first section's titled Map and Protect. So you really can't manage what you don't measure, and in that vein, we really need, just like if you were trying to monitor, declines of plant biodiversity, you'd need to know where plant biodiversity is today to say whether it's been lost or gained. And so this is really what the work of Spun does, and a lot of members of my team do. We use DNA sequencing and machine learning mapping platforms to really map the biodiversity of these organisms for the first time. Get understanding, where's the Amazon rainforest, but for mycorrhizal fungi, for example. Because those teach us, which places, and by looking in places that are degraded as well, so like intensive agriculture, we can begin to understand where we've likely lost components of microbial biodiversity. So using those mapping platforms is really key, because we need this baseline, because we know we're likely losing other components of microbial biodiversity, but it needs to be documented. And so there's a team called Global Fungi in Prague in the Czech Republic have done an amazing job synthesizing more than 20,000 DNA profiles collected all over the world. They've gone through the literature and extracted the data so that we can actually do this kind of work. And we're really hoping that people will begin, doing similar things for bacteria and archaea and all the other life forms that live in the soil and live on the leaves of plants and everywhere else, all these different components of microbial life. Second, we actually mapped not only what we did know, but what we don't know. We made basically an exploration priorities map in this paper. We said, where are all the places where we don't have a microbial sample, and what do they look like with respect to the environment? Are there places in environmental space, unique, places with certain seasonalities of rainfall and temperature that we just haven't captured in our existing sampling? Or are there places that just are really distant and geographically far from where we've ever observed the microbiome? And that's one way we can begin to prioritize, exploration efforts. And so what we see is some things you might expect. We know a lot about North America and Europe and China. We know far less about the tropics. So the Amazon rainforest is massively unexplored with respect to its microbiological diversity. So is the entire continent of Africa, so is Southeast Asia. But there's also some unexpected places. You know, basically the entire boreal forest of Russia, which is a huge ecosystem, has never been really sampled in a way that's accessible to existing academics. And this is really worrying to us because it's actually a predicted hotspot of biodiversity for some of these symbiotic fungi, these fungi that form this partnership with the roots of trees. And they're not hotspots of plant diversity, which is really interesting to us as well. But that's one of these things these mapping efforts can do for us. They can help us identify where we should be focusing our effort to get the most gain out of the resources we have available to do this kind of work. So one thing Spun has been doing is actually going out using these maps we've generated to actually prioritize who we give small grants to. So we give small grants to scientists that live in and are from the places that are massively underexplored, and we basically give them the money they need to go do a sampling expedition so we can refine our picture there, and we arrange for all the DNA sequencing work so that the work gets done up to spec and is compatible with the rest of our data sets. So this is just a really important thing we need to do. We really need, it's incredible that we don't have it. Like, everything relies on microbiological biodiversity. The more we look, the more we see how massively important it is, and yet it's just remarkable how little we've invested in documenting it. So this is really big. Map and protect, we need to know what we have better to really fully appreciate what's at risk and what we're losing. The second part of the paper is really about restoration. So it's one thing to say what we're losing, it's another thing to put it back, though. And we're witnessing a global movement to do large-scale ecosystem restoration around the world. And this was really kicked off by interest from carbon markets. So because of climate change, we know not only do we need to stop emitting CO2, we've given climate inaction to date, we now also need to remove carbon dioxide from the atmosphere. One of the best ways to do that is by restoring natural ecosystems, particularly forests. And now there's increasing interest in doing that, in a biologically and ecologically responsible way. We don't want to just plant like monocultures or a single species of like eucalyptus everywhere just because it grows fast and sequesters carbon quickly. We want to rebuild native ecosystems to the extent that's still possible. So doing responsible restoration is thinking about not just planting trees, but reassembling ecosystems that are biodiverse and capture that native biodiversity that used to inhabit the landscape. But when we plant a tree, we rarely think to quote unquote plant the associated microbiome. So all the other organisms it's really dependent on. And in many environments where restoration happens are ex-agricultural environments. So places that have been intensively farmed for decades and sometimes centuries. And we know the microbiology in those places looks nothing like the microbiology of the native landscapes we're trying to restore. And so one way this is beginning to be done is through what are called soil transplants. So I think to understand this, it's useful to think about what's done in human microbiology. So we know the bacteria that live in your gut have a profound impact on your health. And if you lose certain components of that biodiversity, you can become remarkably sick. But pioneering medical microbiologists discovered that if you take a microbial community from a healthy person, you can re-inoculate and reintroduce microbial biodiversity into a sick person and cure some of these diseases. It's really like microbial ecosystem restoration, but for your body. And that's actually done through a fecal transplant. And that's because the feces that pass through your gut actually capture and retain a lot of that microbiological diversity and can therefore be used as an inoculant. In the ecosystem case, the thing that does digestion is the soil, and the soil is where this incredible microbial biodiversity lives. So we can take small quantities of soil and use it as an inoculant. And so there's some places around the world where this has such a remarkable effect. So one of my favorite example is there's these grasslands in Eastern Europe and Estonia that have been decimated due to mining activity. And when they go to try and restore them, it generally just doesn't work. But if you introduce the microbial community at the same time as you introduce the plants, all of a sudden that restoration becomes possible. And there's these remarkable images of just like with versus without sort of this microbiome treatment, and it's just night and day. And people have done this in other systems and forests. In this paper, we synthesize over 80 experiments where people have done this. And what we see is the results are remarkably variable, but on average, very positive. So on average across studies, we see biomass accumulation and recovery goes up by 64% compared to control, which is a lot. But it can vary from nothing to, huge amounts, 600-700% were the highest effect sizes we saw. And, one thing we need to do next is really understand why that is. We think a big part of it is, again, we need these maps. We need to know what an intact microbial community is so we can successfully sort of rewild it into the soil. And the DNA sequencing work allows us to do that. I think it's really easy, and I've seen this done, is you go and try and do this microbiome transplant, but you go to a forest that seems fine and would seem fine to me or you and intact, but really is regenerated only 20 years ago. And it's just no one knows the previous land use history. And unless you're a forester or someone with some real natural history expertise in the area, you might not get that right. So we need to get a bit more sophisticated in this approach, but where it works, it's remarkable how well it works. And so, what is a forest? Have we really restored a forest if it's missing its fungi? So this is a big thing we need to do. When we think about doing ecosystem restoration responsibly, we think about, the biodiversity outcomes for sure. And not only do we need to be getting the tree species right and the animals right, we also need to be thinking about the microbial life as well if we really, want to do this in a very ecologically responsible way. And I guess I feel like I've been talking forever, so I'm going to try and wrap it up. The last thing we talk about is this opportunity in managed ecosystems. So, food and forest agriculture, represents one of the dominant uses of land on Earth. Something like 50% of the arable land on Earth is in some form of agriculture. And historically, what we've done in agriculture is really an exercise in reductionism. We identify high-performing plant species and then strains of those species, and then we selectively breed them, and now we genetically modify them. And then finally, we plant them out in vast monocultures. So a single plant species as far as you can see. And to be clear, this has produced, very productive ecosystems. But we're also coming in to understand those ecosystems are remarkably fragile, increasingly sensitive to extreme events and novel pathogens. They're incredibly reliant on chemical inputs we're now coming to understand have really serious consequences. However, we have yet to really begin tinkering and playing and modifying what organisms live in the soil alongside of those plants. And we now have the data and the computational tools and the theory to start taking a different approach. Rather than reducing and trying to create a microbial monoculture below ground, just do the same thing you did in plants. What if we went the other way? What if we leaned into complexity and biodiversity and tried to make these systems reservoirs of below ground microbial life and biodiversity? And by doing so, can we do that in a way that also creates positive outcomes for plant nutrition and yields and carbon capture and all the other services we ask of these ecosystems? And so there's huge opportunity here, right? Like, there's a big movement now in regenerative agriculture asking, in what ways can we construct these agroecosystems such that they can hold on to a bit more biodiversity, create habitats for pollinators and other animals? And I think when we apply that lens to soil biology, there's this super exciting world of possibilities here. So that's really the last section of the paper. And so these are the things we think are really critical to get on people's radar and into the conversation in both, restoration and ecology and microbiology. Is, there's this important work of documenting microbial life on Earth and where it's being lost. And if we're going to make intelligent conservation decisions, we really need to map the world's microbiology so we can protect it. We need to fold this into restoration, both because it's an important component of biodiversity and because it can create amazing outcomes for the recovery of ecosystems as well. And then finally, there's this huge opportunity in agriculture to build biodiversity into the managed landscape and also create, all these outcomes we want to see in the world. And so that's really what we did. I hope some of that makes some sense. But yeah, I'm happy to go back and forth and go a little bit deeper on any part of it.
Wow, thank you so much, Colin. This was a wonderful overview and talk, and it's really interesting. We had a guest speaker. He's a PhD student that was kind of by chance that his supervisor had at some point collected samples from a forest, and then there was a mega fire. He explained the exact definition, which I don't want to go into it. And then they collected samples afterwards, and they found that different fungi make it through those mega fires, others don't, but they don't know yet if that's good or bad. Like, are those that survive good? Will they help to restore a forest, or are they actually maybe even hurtful for the future? So um is there you know is there also a plan, once we know more, to kind of build up resilience for the future with the climate predictions we have? Let's say it will be drier or more humid or warmer, to kind of figure out a way to make those transplants to kind of protect that environmental, like that landscape, that specific one, for the future, kind of prepared. Is there a plan to do that, maybe?
Yeah, wow. That like sort of like assisted migration, but for the microbes. Yeah, that's a great point. I hadn't thought too much about that, but it totally makes sense. And when we're doing ecosystem restoration, more and more we're asking, are these organisms not only going to survive the climate of today, but will they also survive the climate of tomorrow? Um, and we know certain organisms are locally adapted to different environmental regimes, and some microorganisms can even imbue drought tolerance onto plants. So there's a lot of different interesting possibilities here. Um, and something that, yeah, I think hopefully will be incorporated more and more. We don't have we're not doing that yet, but it's definitely on our radar.
Is there also people that are working on bioengineering, maybe create a network that sucks up more water and kind of stores it for plants? All this, it's kind of still in the same. Or do you think that's a bad idea because we just don't know the outcome of bioengineering those, the microbiome, basically?
Yeah, I think the most interesting and exciting work in microbial bioengineering is identifying ways we can work with not just like one or two super high performing species, but actually using whole communities of organisms. Um, and asking, can we create both a biodiversity outcome and an outcome for the thing you're trying to engineer for? So we recently founded a company called Funga to translate some of this work into climate and biodiversity action. And where we're focused first is in forestry, actually. So um, we're starting the loblolly pine forests of the southeast United States. This is one of the biggest sort of agricultural crops in the country. There's 30 million acres of these pines. And we're asking, can we identify communities of native and biodiverse fungi that naturally associate with these trees that are also linked to productivity and yield outcomes. And then by doing so, can we essentially do, you know, below ground microbial rewilding and in the process create productivity and a climate outcome? And so it's definitely a form of engineering, but it's placing sort of the biodiversity and complexity component at the center. And I think, there's opportunity to take that kind of approach in so many different areas. Um, and I think that's really exciting. There's definitely also groups historically and now even more so today who do the traditional ag thing, identify some high performing fungi, one species, and then apply it everywhere. And we really worry about that because there's this huge potential to sort of just repeat the mistakes we've made in above ground agriculture below ground. So I think there's a lot of opportunity here to just fundamentally rethink how we do that microbial engineering in a way that, doesn't over engineer for one outcome at the expense of all the rest.
Yeah, thank you. And my hope is that, that will be the case, to focus on diversity. And I kind of have a little bit more hope because of the technology that are available nowadays. Do you think that there's more hope because we just have the ability to go through more data sets on a larger scale and make models with more, higher complexity levels? And so do you think that was the reason why people before, yeah, did this, quite of limited approach? But is there hope because of, us going through more data, having kind of machine learning and all of this available, that, maybe hopefully a lot of groups will have a more, complexity scale approach?
Yes, yes to all of it. I think so. I think, if you think about the things that enable this, like, we couldn't even collect, the data at a meaningful scale 10, 20 years ago, because we either didn't have the technology or it was too expensive to roll out at scale and do frequently. Then, even if we did have the data, it became a compute problem once you had that much data, because one observation of the microbiome is like, thousands of species frequencies, and that is just like a few megabyte. It very quickly becomes enormous amounts of data. And if you want to do this at a large scale with lots of repeated measures, it becomes insurmountable with the tech we were using even 10 years ago. But that's completely changed too. And that technology, in theory, and new computing algorithms allow us to take sort of different computational approaches than we've ever been able to before, that allow us to see signal a lot sooner in a lot more different ways and actually explore the full sort of breadth of what's in these incredibly rich biodiversity profiles. So yeah, I'm definitely hopeful as well. I don't think this would have been possible, even in the near past.
I want to thank you for that answer, and for answering my questions. Kiko, I wanted to give you an opportunity to ask before I go on and on. So thanks, Kirko, for coming, and ask away. Maybe he stepped away from the phone. Okay, I'll keep asking. Are currently also governments interested in your work? I would hope so, but I don't know. So because I would imagine that this approach would be kind of an interest in securing food resources for the future, and also for national safety, you know staying more resilient. So yeah, thanks.
Yeah, totally. So government funding agencies are absolutely funding research into the ecosystem microbiome in agriculture and beyond. Some have funded sort of real large scale sampling programs. Australia comes to mind specifically, so is China. So as you know, the EU has an incredibly well done. They basically added this component of microbial biodiversity to this incredible soil survey they do called the Lucas Soil Survey. And so, yeah, we're seeing more and more uptake there. We're seeing more interest on the philanthropic side, which Spun engages with. Um, so yeah, definitely a lot more government interest than really I think has ever been in environmental microbiology, at least biodiversity science and microbiology before.
Interesting. I'm glad to hear. I remember when I did my first postdoc at the Marine Biological Laboratory on Cape Cod, the co-director of NSF was there to give a talk what we should do for grants and how we should pick our career. And he basically said, just switch to plant physiology and related to agriculture. We were all kind of, in different fields. He said, oh, you can do this in the lab. Just switch because that's where all the money will be.
Oh wow.
I don't know how that is really true right now, but that's the advice he gave. So maybe NSF is a place we should ask for funding. And welcome, Ayab and Philip. Thanks for coming. Yeah, just ask your question. Thank you.
Hey, good morning, everybody. I just have a quick scan at your research, and it's amazing. Just a quick question: I'm I'm trying to figure out how this is gonna work out. Did you ever consider like a species interaction, like if you have this strain of anaerobes versus fungi versus what the Clostridium? Uh, I guess microbiome. I'm referring to a human gut. Thank you.
Yeah, so there's more and more work on sort of how the co-occurrences of species, so not just the presence of single species, but the presence of two or more species in combination is predictive of outcomes versus just the individual species one at a time. What we find, most of my work is in forests, and we've done a similar work to the Human Microbiome Project, but instead sequencing forests and looking at forest health outcomes. We find the best predictors of forest health are almost always community level predictors. So predictors that have information not about the presence of all of the organisms in the sample rather than focusing in on an individual species or strains. And so it's likely that's because of these interactions you're describing. And I believe, that's been the challenge in scaling up human microbiome work, right? We know these low-tech approaches can work really well, fecal transplants where you move the whole system, but developing sort of custom probiotics where you add just a couple of species has proven challenging. And one leading hypothesis is because they're too simplistic, and they need all those co-occurring species to really do well.
Yes, I'm very aware about the stool transplants in the 60s. And I think, yeah, you're right. You can do it like a simplistic approach because I think you have to match like the growth curve, like the saturation decline, raising and how they interact with each other in a specific environment, either a forest or a human colon or a human gut versus any other environment. All right. Yes, that was great. Thank you.
Of course.
Yeah, thank you for that question. And I wanted to give a follow-up comment that the diversity in the human microbiome is also declining. Do you think that's related to lifestyle changes that we don't, I don't know, kids don't play in dirt anymore. We keep sterilizing them, and also the dirt they play in, it's probably not diverse anymore in cities and so on.
I'm not an expert in human microbiology, though I do love to draw analogies to it. But there are results showing like babies born via C-section, for example, have measurably different communities of microbes in their bodies than those not. They think it's because one of the first place you're basically sterile inside the womb, and the first place you experience microbiology and have it colonize your body is when you leave the vaginal tract. So that's a colonization event that, for example, some people don't get now because of technology. And so, doctors are experimenting with doing things like vaginal swabs of babies when they're born via C-section for this reason. We also know like eating dirt is just like a thing young kids do, which is a really interesting behavior and maybe linked to stuff like this. But also, yeah, we don't spend the same amount of time outside, and we also have sort of massively designed our landscapes and our built environment in a way that's like focused on sterility and killing everything, rather than trying to think about like how do we make sure we have microbes here but the right microbes. So yeah, I mean, I think so many different things about the way we live has massively affected sort of this really important component of our biology and our health that we haven't really appreciated until recently.
Yeah, thank you. And Philip, you had the question. Thanks for waiting.
No, thanks, Katarina, for hosting the space, and thanks, Colin, for sharing your experience and expertise. Yeah, think about this recent Living Planet report by WWF, which states that the planet has lost 69% of its wildlife population sizes since 1970 over the past 50 years. So population sizes of animals generally on average have shrunk by almost 70%. Some areas that's much worse. And, they surveyed more than 5,000 animals, birds and reptiles and fish, etc. I mean, in some places, like I said, this impact is much worse. But I'm wondering how this dumbing down of diversity impacts a microbial life in the soils, because I would, that saying, as above, so below. So the diversity that you see above is a kind of a reflection of the diversity that you see below. So I'm just wondering if you can link those two, the decline in animal life and then the decline in the microbes. And then my second question relates to forests. And I'm sure you're talking about real forests when you talk about forests, because when we talk about forests here in South Africa, where I live, it's industrial monoculture timber plantations of mostly invasive alien species like pines from Mexico and eucalypts from Australia. They are maintained and managed as the mother of all monocultures. I mean, you go inside there, there's no sounds, there's no insects, there's no birds, because there's no food for indigenous animals to eat. So I'm wondering what the impact on the soil microlife is by this dumbing down of diversity. And if you can link that to the long-term sustainability of the soil, can we continue with this model of forestry without suffering serious soil and nutrient deficiencies? Thanks a lot for the opportunity to ask.
Yeah, I love both those questions. So the first one, how can declining wildlife populations actually impact what's going on in the soil? In so many different ways. I mean, we know wildlife has these huge effects on which species live and die in a forest, both above ground, but also they can be really important dispersal agents of microorganisms. So, people have observed like certain plants in certain areas were only colonizing where feral hogs also had range and habitat. And the reason for that is because many of these plant species couldn't successfully establish and grow to maturity until, basically the symbionts they needed arrived there as well. And those symbionts were really dependent on these feral hogs for moving around because they dig in soil and therefore move soil and all the organisms that live in the soil around. So, this is pretty understudied. There's not a lot of research on it, but it's just really hard to imagine, given the ways we know animals interact with the soil and move things around, like that it's not having an impact on the biodiversity of what lives in the soil. As for intensive timber plantation, so yeah, around the world, especially in the southern hemisphere, a lot of exotic trees are planted, particularly pines and also eucalyptus, just like you described. If you look at the micro, so I focus on these root fungi. If you look at the diversity of root fungi on like those exotic pines, it's incredibly simplified. So radiata pine is a big one normally, and its native habitat, it's one of the most planted trees in the world, but its native habitat is like this really tiny area on the California coast. And if you look at the number of species, living in symbiosis with its roots, like any one tree easily has like 50 to 100 different organisms there. If you go to these exotic radiata pine plantations in Australia, for example, you see something like 5, maybe 10 species. And so it's this incredibly small, subset of what's naturally there. I wouldn't be surprised if the same things were going on with the eucalypts in South Africa because their native range is in Australia. And then finally, because they're housing these microbes, they're displacing a lot of other microbes that would normally partner with the native vegetation. I think, especially in forest, we have this revolution going on around these concepts in regenerative agriculture. Can we do agriculture in a more sustainable way that's less resource use intensive, that builds soil environments, that creates climate and biodiversity outcomes, or is at least more compatible with biodiversity? And I think we really need to bring that same sort of thinking to forestry. Can we do forest? We in many ways, timber's not going away. In fact, the built environment, like we want to get concrete and steel out of it because they're so greenhouse gas intensive. And if we can build with more timber and less those things, that could be a win. But can we do that in a way that's far more aligned with the biodiversity that inhabits our planet, and do that in a far more sustainable way? We need to really rethink, the single factor optimization we've done up until this point in forestry. Katerina, I have to jump off the phone, but I just wanted to say thank you so much for the opportunity and your work and the chance to just share this research with a broader audience. We think it's so important to share and have people hear about this, and work like yours gets that done.
Well, thank you so much, Colin, for taking the time, and thank you for doing this work, for using your brains to do this and not, I don't know, work on Wall Street or something. So that's also,
Katrina, I wonder if I can have time for one more question to Colin. Colin, would that be right?
I don't know. Maybe you can send me the question and I can ask Colin later on, because he's really
Okay,
busy
super.
right now. He is also planning to give a TED Talk, so follow Colin on Twitter, and there you will get updates, and I'm sure Colin will be happy to answer questions later on, and maybe Colin comes back.
Yeah,
One
happy
day
to do
next.
some answers in writing too.
Okay,
Thank you
perfect.
so much, everyone. It's been awesome.
Okay, wonderful, and good luck for everything. I hope you get all the funding you can get, and you should, people should fund you and support you the best they can. So good luck for everything, and thank you again.
Thank you. So long, everyone.
And we have one room this week. We had to reschedule a room that was supposed to be tomorrow. And it will be on Friday. It's a light schedule this week because in the US it's Thanksgiving week. So it's kind of a busy week with traveling and family. So Dr. Djakovic on Friday will talk about the coexistence of modern humans and Neanderthals, how they lived together quite closely, actually, for really a long time. So it will be a really interesting room, going back to our origins and coexisting with other species. So if you think that's interesting, join us again, and then next week we'll have Dr. Sergey talking about black widow spiders and how he found that they have a really good spatial memory of their prey. It's really interesting how he did that, because usually we do this type of work in mammals, so it'll be really exciting. And Dr. Feldman will talk about breathing patterns and influence on emotion. Like, he has been doing many years research in how meditation affects our brain and our behavior and so on. So he published books. Like, he's a really wonderful researcher in that area, and I feel like during the holiday season it's especially good to keep our emotions at bay. So yeah, I think that will be interesting. And then we'll have Dr. Cohen talking about autonomous micro robots with artificial brains, which will be really interesting. Will have many implications for future technology. And Dr. Simek, ancient art, cave art discovery with 3D photogrammetry. So he will talk about this technology and about the research and applications of that technology in archaeology. So I hope you enjoyed this room, and to hear you all back soon. And thanks for the questions. Thank you, everyone. Close the room in 3, 2, 1. Bye, everyone. Thank you.